is a very widespread eastern North American forestry that has a lot of economic and ecological importance. So acorns are an important food source to furniture, the wood being important in making furniture. So over time, after the Pleistocene, oak trees migrated very rapidly from south to north, and there is this massive climatic gradient. So we assume, with natural selection theory, that there would need to be some sort of adaptations necessary to deal with different climatic stresses, such as cold and drought. And that is specifically what I am looking at here. So essentially, I was able to amplify candidate gene, or regions within candidate genes in Quercus rubra. So the process that I did this by was finding in the literature candidate gene sequences and identifying candidate genes in model species like Arabidopsis, thaliana, thalecress, peach, Populus, apple, and using blast sequences of those, was able to blast, which is a way to pull down similar sequence, blast it against a Quercus rubra transcriptome, low resolution Quercus rubra transcriptome, and then be able to try to target exon-intron-exon regions. So we would hopefully have enough power to differentiate natural selection from genetic drift, which genetic drift is random processes. So essentially, the results here show, I present here 5 out of 13 candidate genes that I was able to amplify reliably. And as you can see here, figures 2 through 6 are alignments of these genes, the regions in Quercus rubra to these other model taxa, be it peach or vitis, which is grape, populus, poplar. And in these figures, with the exception of this one where it turns out the intron is most likely deleted in oak, you can see that there is tight alignment, very nice alignments in the exons, where I have little black lines here to indicate the exon-intron boundaries. And then there is a lot of variation within the intron. And then when you hit the exon again, the sequence tightens back up. So, and with aligning these across taxa, we can actually reasonably assume that we have orthologous genes, or genes that we know that have a similar, I guess, ancestry and function. So the significance of this is then I'll be able to use these genes to sequence them in individuals across the range from south to north, and to hopefully see a signature of natural selection for cold tolerance and also drought tolerance, because there has been some recent work showing that drought and cold tolerance are very much intertwined.